Two rectangular pieces of acrylic are laser cut and then assembled to form the stereotaxic clamp, which serves to hold the injector array tightly with respect to the stereotax. A syringe pump is loaded with syringes connected to polyethylene tubing. For a desired brain circuit, a mill is used to drill holes through which fused silica tubing is inserted, thus forming injectors. The injectors are reversibly mounted in the clamp and then trimmed to target the brain circuits of interest. Each injector is attached to a single polyethylene channel. Preparation of the system for use involves filling the system via the syringes with silicone oil and then loading fluidic reagents into the injectors. The parallel injector is then inserted into the brain targets and reagents infused.
Hi, I'm Stephanie Chan from the Synthetic Neurobiology group at MIT. Up till now, expressing genes in spatially complex brain circuits has required either the slow and expensive creation of transgenic animals or the painstaking process of viral injections, which must be done one site at a time. Today, we're going to show you how to build and make use of a parallel injector array that can deliver fluids to complex three-dimensional patterns in the brain. We use this array to rapidly make circuit-specific transgenic mice, delivering viruses in parallel through the set of injectors to hit all of the targets at once. Our injector can be used to deliver any fluidic reagent to a complex distribution of brain sites. Our goal today is to deliver viruses that encode for light-sensitive proteins so that we can turn on and off